Marfan syndrome is an inherited disorder caused by genetic mutation of fibrin-1, which predisposes to later aortic aneurysm. Marfan syndrome is associated with increased TGA-beta signaling, but it remains controversial whether TGA-beta activation is a complete mechanism. On the other hand, little is known about the mechanism by reactive oxygen species which promotes aneurysm in Marfan syndrome. We observed a significant increase in loss generation in asthenic aorta Marfan syndrome mice holding the fibrin-1 mutation, which was associated with increase in protein expression and enzymatic activity of endothelial xanthine oxidoreductase (XOR). We already showed that fibrin-1 mutation led to abnormal mechanism sensing in vascular endothelial cells, and it mediated aortic aneurysm progression by endothelial XOR activation and loss generation in Marfan syndrome mice. Besides, we observed upregulation of XOR in macrophages infiltration aortic adventitial Marfan syndrome mice. To elicit the role of XOR in macrophages, we inhibited in vivo function of XOR in macrophages by lysosome-mediated disruption of XDH gene encoding xanthine dehydrogenase in Marfan syndrome mice. This slide shows representative ultrasound images of aorta for wild type and Marfan syndrome mice and macrophage-specific XDH knockout Marfan mice. In macrophage-specific XDH knockout Marfan mice, aortic dilation was significantly suppressed as compared with control Marfan syndrome mice. And next figure shows histological changes such as aortical thickening, degeneration of elastic fibers were significantly attenuated in asthenic aorta or macrophage-specific XDH knockout Marfan mice with a significant decrease in loss generation. And also, these improvements were associated with a significant inhibition of activation of ERK1 and 2 and SMAD2. However, adventitial macrophages infiltration was not attenuated in asthenic aorta of macrophage-specific XDH knockout Marfan mice. Interestingly, macrophage-specific XDH knockout Marfan mice shows downregulation of XOR not only in macrophages but also in vascular endothelial cells. In conclusion, these results suggested a vicious cycle of XOR-derived loss generation between vascular endothelial cells and adventitial macrophages aggravates the pathogenesis of aortic aneurysm in Marfan syndrome.